Thank you very much, and welcome everyone. I'm Tamás Nagy, and during my master's thesis, Tamás Nagy was my supervisor, and we were working with transmembrane proteins, and we ran molecular dynamic simulations to better understand their dynamic behavior. And at this point, you may ask what and why. And well, cells in your body have membranes and they metabolize. And the membranes separate the intracellular space from the outside environment. And during metabolization, the cell should carefully consider what goes through its membrane. So there are many ways of this transport could take place, but the two main categories are, the first is passive transport, which means that the substrate goes along its electrochemical gradient. And the second one is active transport, in which case some energy is required to move the substrate against its electrochemical gradient. And this energy can come from ATP hydrolysis, and ABC proteins are a good example for this kind of transport, where ABC stands for ATP binding cassette, and this is basically a super family of proteins. And inside this super family, you can find ABCG2, which is the second member of the G subfamily. And this protein is responsible for many functions in the body, and an important one is the multi-drug transport, which means that it exports a wide variety of cytotoxic molecules from inside the cell into the extracellular space. And this behavior helps defending the brain, the embryo, and the newborn children, because it is expressed in the blood-brain barrier, the placenta, and the breast. But this ability can be detrimental too, because cancer cells can develop multi-drug resistance, and which means that the drug concentration is lowered in the cell, and this hinders the effect of chemotherapy. And then other function is uric acid transport, and the specific mutation of G2 makes it unable to transport uric acid, and thus uric acid concentration will rise in the blood, and it can lead to gout, or in Hungarian. So here's the structure of ABCG2. The functional structure of the protein consists of two polypeptide chains, which two protomers that together form a homodimer. And here you can see two different conformations for this specific protein. On the left side, the invert open conformation, which enables the substrate to enter the protein and allows ATP to bind. On the right side, you can see the closed conformation, which occurs while substrate exits into the extracellular space. In green, you can see the nucleotide binding domains, NBDs, and as their name implies, they are responsible for ATP binding and also other tasks. And in gray, you can see the transmembrane domains, which are forming the translocation pathway across the membrane. So inside the transmembrane domains, we have site 3, or the main binding pocket. This location is targeted by many studies, and it plays a key role in substrate recognition. So along the path leading to site 3, we suspect the existence of site 2 on both sides, and which may act as a preceding filter to substrate recognition. And even though we have these structures, we know little about the dynamics of the transport mechanism and substrate recognition. So to discuss this further, I'd like to propose an analogy to the basic dynamics of the process. So here you can see a canal lock where this shift corresponds to our substrate that we want to move out from the cell. And as it enters the binding pocket here, it hydrolyzes ATP, the conformation of the protein changes, it becomes closed, and the energy level rises. And the substrate, or sheep, is released into the extracellular space. Okay, and at this point you may ask, all right, but why are you here? And well, we are investigating organic macromolecules, and we want to observe their dynamic behavior on atomic level resolution. So most experimental methods are not suitable for this kind of observations because, for example, the cutting-edge cryo-electron microscopy only creates static images because the protein is frozen. So, but we want to learn about the dynamics. And for this, we are running molecular and dynamic simulations. So here you can see one of our systems, simulation systems, which contains some water, the extra and intracellular space, separated by the membrane in green, and you can barely see our protein in blue. And this system consists about 200,000 atoms, and the computer solves Newton's equations of motion. But it takes a lot of time because of the many atoms and the many interactions. So actually, 500 nanosecond simulation took 3 weeks with 4 high-end GPUs. And during these simulations, after every other step, we write the state of the system into the disk. So we end up with a trajectory containing all the coordinates at the given time. And this is the data that we can analyze. So you can see a movie from this trajectory. All right. So we have existing experimental data about the regulatory effects of cholesterol. Basically, the cholesterol presence in the membrane increases the activity of ABCG2, but the exact mechanism of this activation was not known. So to get more insight, we run molecular dynamic simulations for cholesterol-containing membranes and membranes without cholesterol for simulation for each case. And on the following diagram, you will see that the locations of the intracellular ends of the helices in the membrane. As you can see from this little picture. And here each cross corresponds to the initial position of one helix end. And the top diagram shows the cholesterol-containing membranes, and the bottom one is for simulations without cholesterol. And then this heat map shows how frequent was a given coordinate during the time span of the simulation. So brighter red color means that the helix end spent more time at a given location. And what we observed is that in case of the simulations with cholesterol, some central helices were much more likely to close. These helices are marked with a red cross, the initial position. And to quantify the results, we plotted the distance between these two central helices. And then this is what you can see on the right side. As you can see, in the case of cholesterol, the central helices were much more prone to close. Then the distance was much more likely to decrease between time, as the time increased. And in contrast with the membranes without cholesterol at the bottom, where we can see they were even opening in some cases. So we concluded that cholesterol exerts its regulatory effect by helping the structure to close. And to allude back to the canal lock analogy with the sheep, cholesterol basically acts like grease that helps the closing of the gates. After that, we ran simulations with uric acid molecules around to observe the interactions with the protein. And during one of these simulations, the uric acid entered the protein. And on the figure, you can see the initial conformation of the protein, but it's important to note that it's not stationary during the simulation. It's moving, but it's only for visibility. And it interacted with some amino acids at site 2, and then went on the translocational pathway as the time increased, and it reached the main binding pocket, or site 3. But it wasn't able to go through into the extracellular space because the energy barrier for this was too high. So then it moved towards side 2 of the other side, and that's where its journey ended. And the simulation gave us some key insights about the first part of the pathway, but to gather more information about the second half, we had to overcome the energy barrier. So for this, we used metadynamic simulations. And during metadynamic simulations, we define a reaction coordinate, for example, a distance between specific amino acids and the substrate. And then we monitor the value of this reaction coordinate, and if the system stays too much time to give a reaction coordinate, we artificially increase the free energy level of the system at that point to make the system move. And you can see these steps on the right side of the image, on the right side, where the reaction coordinate is in the horizontal axis and the free energy level is on the vertical axis. So you can see that the system is trapped at the local energy minimum and moving away. And this method is good because we can measure how much energy is needed to get over these energy barriers, and thus we can get information about the system. So we've run metadynamic simulations for the inverse closed conformation of the system, and we defined the reaction coordinates as the distance between the substrate and some amino acids of the protein. And we've run 6 simulations with this system, and we were able to observe the substrate exiting into the extracellular space in all 6 cases by interacting at various locations along the way. And we also measured the energy requirement of the translocation, and in 3 cases out of 6, it was below the ATP hydrolysis energy level. So this implies that the results are realistic. So in summary, we determined the exact mechanism of cholesterol's laboratory effect, and our results show that it helps the closing of the protein structure. And we also determined key points, binding pockets along the translocational pathway involved in the substrate recognition. And as you can see, this kind of research requires a large amount of computing capacity. So I'd like to thank Wigner GPU Laboratory that they provided their GPU clusters to aid the research. And if you are interested in the topic, you can also read our recently published paper or visit hegelog.org. And I'd like to thank you for listening, and I'm here to answer your questions. Thank you.
and thank you for taking the time. Anybody outside or inside? Questions? Time is pretty much over. I would have a question in relation to the slide 13. So, I mean, you know, we are dealing with giga and tera electron volts, which are a bit larger energies in some sense. So, you said on the middle of the figure, there is this number of simulation versus the free energy in kilocalorie per mole. So when you presented that you have this potential with a certain forms, so is this within the uncertain? So I mean, I cannot really get the energy range. Okay, so I see that between the different simulations, you have like 5 kilocalorie over mole energy difference, and of kind of how do I say, uncertainty or so this somehow was not clear for me. I know the question is a kind of silly, but I didn't get this point.
So this is just for presentation purposes. It's a general graph or image. I said so. It's not related to our system. It's just to visualize what's happening during the multi-dynamic simulation in general.
Okay. Another question is we have heard several machine learning or based studies today and even tomorrow. So don't you think to use such techniques could be order of magnitude higher result, produce much more faster results than the standard methods? Are such things available for these kind of problems?
Well, the problem is that to get our data, we had to calculate, so run the computers for a long time to get our basically raw data that we can analyze later on. And I think that machine learning algorithms work great when the data is plenty.